only they click to join meeting na and they continue on the browser cancel continue on the browser আসলে কোনো অসুবিধা দরকার নেই হ্যাঁ আসলে এসি করে এত মাস দেখে ওইটা তো এসি ওটা আছে বলতে না ওটা এখন শুরু হয়নি বাবা আমাদের নেটওয়ার্কের প্রবলেম চলছে বুঝলে
না না তুমি করনি বলে নাকি আমার তো চলছে
ওটা করিনি বলে আসছে নাকি মনে হচ্ছে
দাঁড়া একটা দেখি শোনা যাচ্ছে
না ছাড়তে হ্যাঁ ছাড়তে দেখাচ্ছি তো দেখাচ্ছি না শুধু করতে লাগে স্ট্যান্ডার্ড করে
ওটা তো লাইটিং এর সাইজ টাইটেলটা দে
বাবা
এটা ব্যাকটেরিয়ার
দাদা তোমার মিউট করো
I can record.
The microphone is broken. There's nothing.
So does that mean that you have to stay close to here, or is it fine?
I think now, yes. Now we hear something.
Okay. Okay.
Okay, so back to full screen mode. How do you do that?
Yeah, and I hear something. Yeah, somewhere. Yeah.
Someone's making here.
No,
Yeah, I can.
you can do that. Okay.
Okay. Well, thank you for that wonderful introduction. Thank you all for being here. It's an incredible honor to get to be here and to learn about all the exciting things that are going on here at Imperial College. It's been a few decades since I was last here, so it's a pleasure to be back. And it's a real pleasure to get to share some of the work that we've done over the past few years. So as Anna mentioned, my group studies, broadly speaking, the soft materials and living systems. We had, and transport processes, for example, in porous media, we had two incredible postdoc talks previously talking about various kinds of problems in fluid mechanics, which were really fascinating. And then I heard also that Lewis Hamilton came here yesterday. And so these are really tough acts to follow, but I will do my best. Today, I'll tell you about some of the work we've been doing studying how bacteria move around in complex spaces. And so it's kind of like Formula One, just at microscopic scales. So hopefully it'll be as fun. But first, before I dive into the subject of today's talk, I wanted to give you all a brief overview of what we do in my group as a whole. So broadly speaking, we study the dynamics of squishy things, so soft materials, and living systems and active systems in complex spaces. And so when I say squishy and living systems, I mean a broad range of different classes of materials, ranging from fluids and immiscible fluid mixtures to polymer solutions and gels to colloidal dispersions, all the way to communities of cells. And as a community, we've learned a tremendous amount about these different systems, primarily by studying them in bulk, uniform, idealized environments in the lab. And that's given us a really critical understanding of the scientific principles that underlie their behavior. But in nature, in many engineering applications, we often encounter them in more complex spaces, in spaces with structural disorder and confinement and crowding, chemical heterogeneities, mechanical heterogeneities, fluid flows. And these are all factors that can profoundly change how these different systems behave. And disentangling those interactions is what we focus on in my group. So what are some examples of crowded, complex spaces that we're all familiar with? Well, one example is very familiar to all of us, and many of you study these kinds of complex spaces. It's the ground beneath our feet, right? When you go outside and take a walk, soils, sediments, subsurface formations, these are all examples of disordered porous media right beneath our feet. This, for example, shows you an X-ray CT image of soil, where you can see these brown particles are the solid mineral grains that are all packed together and house this tight and tortuous pore space through which fluids and solutes and even cells move and organize themselves in a range of different processes. And so disentangling those interactions is what we focus on, motivated by their importance in applications in energy and environment and sustainability and biotechnology. There's another example of a complex and crowded space that you're all also very familiar with. It's the tissues and gels in our bodies, right, which are often made of porous, disordered macromolecular networks. And so I always love to show this picture in my talks, mainly because it gives me a lot of joy. This is my daughter, Aiko, who is 4 years old now. And you can see here, she's a happy baby. She's smiling, and she's doing what babies do best. She is slobbering all over the place, right? And not only is this process incredibly adorable, but it's incredibly important for her health because mucus is an example of a macromolecular network that's secreted by our bodies, and its mesh-like structure helps to regulate how it serves as a biological barrier against inhaled pathogens and particulates. So again, we're interested in understanding how fluids and solutes in cells navigate these crowded spaces inside our bodies. And so, I'm primarily trained as an experimentalist, and as an experimentalist, when you try to study what's going on inside these crowded spaces, immediately you're faced with a challenge, which is that many of these media are opaque, right? If you take a handful of soil and you try to look inside of it, it's challenging, it's opaque. You can't actually see what's going on inside, right? Tissues and gels similarly. And so what we do in our work is we design experiments that try to overcome this challenge, inspired by this wonderful quote by the American baseball player Yogi Berra, who once said, "You can observe a lot just by watching." What we do is we try to develop experimental model systems that have controllable structures and chemical properties and mechanical properties that can recapitulate many of these complexities inherent in nature and engineered media, structural disorder, heterogeneities, etc., but that are completely transparent. And so we can spy on what's going on inside of them using, for example, confocal microscopy. And so guided by those experiments, we then try to develop mathematical models to help generalize our results more broadly. And so there are three areas that we focus on in our work. One area is in tackling this classic challenge of trying to predict and control how fluids flow through porous media. Many of you also study similar questions. Right here, for example, you're seeing a movie of an oily contaminant, that's the black stuff, invading a porous medium that's saturated with water. That's the bright regions, right? This is deep inside one of these 3D porous media. And we take this movie by making our own model 3D rocks that are transparent and that have controlled structures and properties. And so as you can see by looking at this movie, the oily contaminant, as it displaces the water, it doesn't just smoothly go from left to right, but it navigates the pore space using this tortuous fingering pathway, right? And by taking movies like this, we've been able to develop a deeper understanding of the physics underlying these kinds of flows. And so, for example, we've studied how structural heterogeneities and chemical heterogeneities that are inherent in many natural and engineered media can profoundly reshape these flow behaviors. We've studied how colloidal dispersions navigate these pore spaces, how the little particles, as they squeeze through the pores, they can get stuck in tight spots, they can redirect the flows to other pores, they can get pushed through by hydrodynamic forces and keep spreading over large distances. By visualizing these coupled dynamics, we've been able to develop mathematical models to describe colloidal spreading over large length and time scales in our environments. We've also been studying how viscoelastic fluids flow through these porous media, right? And in these cases, you have new physics that arise. For example, because the polymers in these fluids that are often used in industry are stretchy, and it takes some time for the elastic stresses to relax back in the flow, those stresses can build up, and above a threshold flow rate, these flows can become chaotic, even though these flows are always at low Reynolds numbers. And as you typically learn about in fluid mechanics, right, at low Reynolds numbers, you expect the flows to be linear, to be smooth, stable, unchanging in time. And yet, polymer elasticity can impart new features, can generate chaotic behavior in these flows. Right here, we're looking at a single pore in a larger 3D porous medium. We're watching the fluid go through, and this looks like turbulence, even though these are conditions under which turbulence typically doesn't arise. Right? And so by visualizing these flow instabilities, we've been able to develop a deeper understanding of when they arise and what their implications are for macroscopic transport behavior. We've also studied what happens when the porous matrix itself can deform, for example, in hydrogels or deformable packings, where you have an intimate coupling between forcing fluid through the pore space and squishing the overall material. And we've studied how to harness that coupling to control the shapes of things, for example, to make self-healing materials that spontaneously heal cracks. We've also studied how these fundamental processes of fluid absorption and swelling, or desorption and fluid release, are altered when these materials are now in confinement, as in many applications. And then the third area that we've been focusing on is using these tools and approaches from fluid mechanics, from chemical engineering, to study living systems, and in particular bacterial communities. And that's what I'll talk about today, right? And the work that we've been doing in this area is inspired by a very simple question. For over 300 years, ever since we first discovered bacteria, right, typically in the lab, we study bacterial cultures in test tubes, right, in bulk liquid cultures, or on petri dishes on flat surfaces. Right? And that's given us incredibly important insights in microbiology. But in reality, many bacteria don't live in bulk liquids or on flat surfaces. They live in our bodies. They live in soils. They live in sediments. They inhabit complex, crowded spaces. And so the question that we've been addressing is, how does that change how bacteria behave, right? And over the years, we found that the way individual cells move is completely different from what we expected, from what the textbooks told us. The way collectives of cells move and chemotax and grow is completely different from what we expected. And so today I'll share some of those vignettes with you. But first, I want to highlight the people who made all the work I'll talk about possible. All the experiments I'll talk about today were started and led by a former postdoc in my group, Datta, who's now a faculty member at the National Center for Biological Sciences in India. Konane Bey is another former postdoc who continued to push those experiments along. She's now a faculty member at CU Boulder. Jenna Ott and Anna Hancock are two incredible graduate students in my group who have continued to push ahead with the experiments, along with Hao Ni Lu, who is a very talented undergraduate who's now going off to MIT for graduate school. Daniel Amchin is another former PhD student who developed some of our simulation capabilities inspired by our experiments, some of which I'll tell you about. He's now a postdoc at Johnson & Johnson. And we've also developed mathematical models to try to describe what we see in our experiments. Ricard Alert is a former postdoc at Princeton who worked with us on that. He's now a group leader at the Max Planck Institute in Dresden. And Alejandro Martinez Calvo is a current postdoc in my group, along with my collaborator, Ned Winkree. And so these are the people who made all this work possible, and all the credit goes to them. So today's story begins with the initial discovery of bacteria over 300 years ago by this person, Anton van Leeuwenhoek, who was a Dutch microscopist. He invented the microscope, and he wanted to play around with it. And so he went to a local pond and took a drop of pond water and looked at it under the microscope. And he reported seeing, for him, among all the marvels he had discovered in nature, the most marvelous of all. He reported seeing many thousands of living creatures, all alive in this little drop of water, moving among one another, right? Many of these were bacteria. He even drew a schematic of this, right? These were bacteria. And so not only was this the initial discovery of bacteria, but ever since their initial discovery, the ability of many bacteria to self-propel, to swim through fluids, right, has been one of their key characteristics. Usually when people quote van Leeuwenhoek, by the way, they stop right here. If you keep reading this letter, it's kind of amusing. He says, my method for seeing these, I do not impart to others, but I keep that for myself alone. So not only was this the first study of microbial motility, it was what I would argue one of the first examples of an incomplete method section. Needless to say, this observation has been reproduced many, many times since in the subsequent 300 plus years, and it's culminated in our modern day understanding of how many microorganisms swim through bulk fluids, right? And so today I'll focus on the case of E. coli, which is a very commonly studied canonical microorganism. We study many other species in my lab, but I'll just focus on E. coli for the sake of simplicity. E. coli and many related bacteria move through what's known as run and tumble motility. Okay, this is what you read about in the textbooks, and the idea is very simple. So each cell body is like a little submarine. It's around 2 micrometers long, half a micron in diameter, and it has these appendages that are known as flagella, right? And these flagella rotate, and when they rotate synchronously, they form a bundle that pushes the cell along a constant straight line path through the fluid at a constant speed, on average for around 2 seconds, okay? And that process is known as a run. And then one or more of the flagella spontaneously spins in the wrong direction, right? And that causes this bundle to splay out. And that hydrodynamic force causes the cell body to reorient. And then the flagella quickly come back together in this bundle, and it pushes the cell along another run in a different direction. And this reorientation process is known as a tumble. And it's effectively instantaneous. It lasts around 100 milliseconds, which is over an order of magnitude shorter than the duration of a run. And so then the cell zips along these runs and tumbles. And what's beautiful about this process is this is a random walk, right? And because this is a random walk, you can use the diffusion equation to quantitatively describe how these bacteria spread out through bulk fluids due to their active motility with an effective active diffusion coefficient given by the product of the run speed times the run length, right? And those are just set by intrinsic cellular parameters, right? And so this is a beautifully elegant picture that's been validated in many studies of swimming microorganisms in bulk fluids over the years. And the question that we had is, well, how has this picture changed when these cells now inhabit a crowded, tight, porous space, right? Not only is this question both fundamentally interesting in active matter physics, because swimming bacteria are model systems of other actively moving agents, but it's also important, for example, in agriculture, if we want to describe the dynamics of how soil bacteria are recruited to plant roots and help sustain plant growth. It's important, for example, in bioremediation, where there's a lot of interest in using beneficial bacteria in the subsurface to sense chemical contaminants, move through the pores of soil towards them, and degrade them in situ. To be able to design those processes effectively, we have to be able to say something quantitative about the dynamics of that process. We hope that these insights could shed light on how infections spread through the body, or alternatively, how to use beneficial microorganisms to deliver drugs to artery spots of the body. And of course, we believe that the insights that we learned from studying bacteria could help inspire the development of new forms of synthetic active matter. So these are all the reasons why we're interested in this question. Ultimately, the question is very simple. We know about this run and tumble process. This is what you read about in the textbooks. How is this altered now when these cells are living in a tight spot? And so typically it's hard to actually watch that, right? To take soil and watch what the microorganisms are doing. But people have come up with models of what could be happening, right? One of the most commonly used models relies on an assumption that was first proposed by the French physicist de Gennes, who suggested that maybe in a crowded space, these cells are still doing run and tumble motion, right? But now, because there's obstacles around them, they bump into walls. And so in this case, the runs are truncated, and the run length is given by something like the mean pore size, right? And so what's nice about this picture is you can still make a quantitative prediction. You can still write down an active diffusivity that's given by the product of the run speed, thought to be unaltered, and the run length, which is now truncated and given by something like a mean pore size, right? It quantifies the intuition that in a dense matrix, the bacteria are reoriented by collisions with obstacles around them, as well as their intrinsic tumbles, right? And so this is a beautifully elegant picture. You can make quantitative predictions, but the problem is that often these predictions don't work very well. For example, when people have performed experiments taking a packing of soil, for example, and you take a suspension of motile microorganisms, put them at one end, measure how long it takes them to get to the other end, and from that you can extract a measure of this diffusion coefficient. The measured diffusivities are often at least 1 to 2 orders of magnitude lower than what you would predict from this very simple picture, right? So we're missing something. And the question is, what are we missing, right? And not only is this an academic question, but it has important practical implications as well. So for example, in bioremediation, right? If I want to describe the dynamics of how long it would take motile microorganisms to move through soil, let's say just over a foot, right? This difference of 2 orders of magnitude in the diffusion coefficient could mean the difference between waiting a month versus waiting over a year, right? So answering this question has important practical implications as well. And so, long story short, what we decided to do when we got interested in this problem is we said, let's watch. Let's make our own model soils, our own model tissues that are transparent, and watch what the cells are actually doing inside of them. And so that's what we did. And so my background is in fluid mechanics and rheology. And so what we decided to do is actually use a trick from rheology that will reemerge later. We use what are known as granular hydrogels, okay, to make our model 3D packings. What we do is we take dry hydrogel grains. So these are cross-linked networks of polymers that really like water, okay, and they're biocompatible. So when you take one of these dry granules and disperse it in whatever liquid cell culture medium that you want to use with the right nutrients and osmolytes for the cells, it swells up, it absorbs all that liquid, it absorbs those salts, those nutrients, until it can't swell anymore because of its elasticity, right? And so now you have this liquid infused porous sphere. And so if you add many of those dry granules to your liquid, well, eventually you get a jammed packing of these particles, right? Like a ball pit for the cells. If you add more of the dry granules when you prepare this, you get a tighter, more dense packing where the pores between the granules are smaller. If you add less of them, you get a looser packing where the pores are bigger. And so just by tuning how much of the hydrogel you add, you can tune very precisely what the pore size distribution of these granular packings is. The internal mesh size of these hydrated hydrogel spheres is around 100 nanometers. We've measured that. They're highly swollen. And what's nice about that is, for the cells, which are much larger, they can't go into the hydrogel. They have to go around the spheres through the pores between, right? They see a solid matrix. But for oxygen, for nutrients, for salts, for the small molecules that the cells use to survive, which are much smaller than that mesh size, they diffuse through completely unimpeded, right? They don't even see the solid packing. And so this platform gives us a very clean way to isolate the influence of physical confinement on what these cells are doing. Right? And so we can map out, for example, the 3D pore space structure by diffusing fluorescent tracers through, and using those diffusion measurements, we can measure a characteristic smallest pore dimension that the cells are feeling in this pore space. So here are 4 measured pore size distributions. The y-axis is logarithmic, right? For 4 different packings. The loosest packing is shown in blue here. As you make them tighter and tighter and tighter, the pores get smaller and smaller and smaller, right? This one has pores between 1 and 14 microns, all the way down to between 1 and 4 microns. And we've broadened these pore size ranges since. But what I want to draw your attention to are these numbers in the top right here. These numbers indicate the mass fraction of the hydrogel polymer that we use to make these packings. Okay, and you'll notice that it's always less than 1%. These granular packings are made of over 99% water. The reason why they form solid packings is because the hydrogel is so highly swollen. Okay? But because they're made of over 99% water, because the hydrogel is so highly swollen, these packings are completely transparent. And so we can then put fluorescent cells inside the pore space and visualize them as they move in 3D using confocal microscopy. And so that's what we did. So when Datta first developed this platform, I said, let's take some fluorescent E. coli. So these are cells that are constantly swimming, right? In bulk media, they just do run and tumble motility. And they're constitutively expressing a green fluorescent protein. So they're always lit up inside their cell bodies. And let's just watch. Are they doing run and tumble motion, as everyone assumes, or are they doing something else? So here are some of the first movies Datta took of E. coli moving in these porous media. So these are at very dilute volume fractions. I just took both movies and put them on the same slide. The cells are all very far away from each other, and these movies will loop. You're watching the cell body as these cells move through the pore space, but they're surrounded by these obstacles. And when Datta showed me this movie, I was blown away. I fell out of my chair because these cells don't look like they're doing run and tumble motion. I told you in run and tumble, a cell does a run and then another run. Tumbles are instantaneous. Here, a cell moves and then it gets stuck. It wiggles around for a bit until it finds the right orientation to get out. It hops out until it gets stuck again, and so on and so forth. Right? These cells are hopping between tight spots of the pore space where they're getting transiently trapped. So this is a fundamentally distinct mode of motility that we call hopping and trapping, by analogy to other forms of passive transport through disordered porous media. And so by tracking the individual cells, we've been able to characterize many aspects of this new mode of motility. I won't go into the details in the interest of time, but for example, we've looked at the mean square displacements, various statistical descriptors of this motion. I should point out that here, all the pores are bigger than the cells, and still the cells are swimming through the pore space. They're not getting squished. They're still trying to do run and tumble motion, but it manifests as this fundamentally distinct mode of motility. And so one feature that we found, again, if you're interested, I'd be happy to chat about this afterwards, by tracking these cells moving through these porous media, is that the way they move, you can actually think about it as being analogous to the way active particles move through disordered energy landscapes. Okay, they get stuck in these little entropic traps until they hop out and get stuck again. And so there's a lot of really interesting physics here. We've been able to better understand this process by looking not just at the cell body, but also the flagella that propel the cell through. So this movie will loop again. I love this movie. You see the cell is trying to move to the left. The flagella are bundled, they're rotating, they're pushing the cell to the left, but it can't get anywhere. It remains stuck for 11 seconds, right? Because there's an obstacle in front of it until finally it gets just the right orientation for the flagella to unbundle, rebundle, and propel the cell through the pore space, right? So there's a lot of beautiful physics here, but long story short, we have a revised view of motility in this crowded space. We no longer have this typical run and tumble process with these truncated runs, but we have a distinct mode of motility, this hopping and trapping, where these cells hop through channels in the pore space. We've shown that that's set by the geometry of the pore space. So you can predict and measure the lengths of these hops. And they get stuck in tight spots of the pore space intermittently and transiently. They get trapped, right? And you can think about that as being an active particle that's stuck in a free energy well. And it needs activity to escape that trap. Again, you can predict and measure the dynamics of that process. This is still a random walk, but this is a random walk with pauses, right? And so what's nice about this hopping and trapping picture is you can come up with a new prediction for this long time active diffusion coefficient of these cells, right? Where the characteristic length scale is given by the hopping length, that's set by geometry. We can predict and measure that. And the characteristic time scale is set by this trapping duration, which again, we can predict and measure. That's set by the competition between confinement and activity, right? And by putting those quantities together, we came up with a new prediction for this long time diffusion coefficient of these cells. And so let's test that. So we tested that by taking a dilute spherical bolus of these cells, embedding it within one of these hydrogel packings, and measuring how long it takes for these cells to spread out over long time scales, right? And large length scales at the periphery. And so 3 measurements in 3 different packings are shown on the y-axis here. Our simplest possible prediction from this hopping and trapping picture, right, is shown on the x-axis here, where we just take the mean hopping length as the characteristic length scale, even though there's a complete distribution of these length scales, and we just take the mean trapping duration as the characteristic time scale, even though there's a power law distribution of these trapping times, and there's a contribution of the hopping duration to this time scale. Nevertheless, in this simplest possible hopping and trapping model, we get good agreement with our experiments within a factor of 3. That's what this line is. And if you're not impressed by a factor of 3 agreement, I'll contrast that with a typical run and tumble model that people used to use, using the mean pore size as the characteristic length scale. That prediction for the diffusivity is nearly 2 orders of magnitude off. So that line would be, far out into the next room over there, right? So this hopping and trapping model gives us a much better way of predicting quantitatively the long-time dynamics of how these cells explore crowded spaces. And in follow-up work, we've used continuous time random walk simulations to further improve these agreements using those full distributions that I mentioned. We've also generalized this to other forms of active matter. So we've studied other species of bacteria in my lab and we found similar behavior. And in a collaboration with my colleague Howard Stone and Hartmut Lowen, we constructed simulations of active polymers. So these are polymers that can self-propel. And we tested how they move through crowded spaces, both in 2D and 3D topologies. And independent of the topology, we found that they still do this hopping and trapping motion. And by analyzing that hopping and trapping, we're able to come up with a geometric criterion for determining when this active diffusion coefficient is maximized. So when the spreading of these active particles is maximized in crowded spaces. And I'd be happy to chat more about that if you're interested. But everything I told you about so far was for the case of isolated individual cells, right? But in nature, in many engineering applications, we often care about more concentrated multicellular groups. Right? And so the next question is, well, how do more concentrated populations spread through crowded spaces, through crowded media, right? And in fact, there were theoretical predictions. You know, people know that active swimmers, just in bulk liquid, can coordinate their motion and they can move collectively in groups. And for example, they can flock and swarm and do interesting things. But there were theoretical predictions that in a crowded space, because now you have obstacles everywhere, the particles are bumping off the walls and their ability to coordinate their motion diminishes, right? And so there was a prediction that collective behavior should not be possible in these crowded spaces. We wanted to test that. And so how do you test that? Well, first, you need to be able to line up all the cells at the same starting line and watch them go. And experimentally, that's very challenging until we realize that our granular hydrogel packings give us a really nice way to do this, okay? And so again, we're using a unique feature of the rheology of these packings. They're yield stress solids, which means that, you know, I told you these are like ball pits for the cells, right? So imagine going to a ball pit and plunging your arm into it and dragging it through. What happens? Well, the balls in the immediate vicinity of your arm get pushed aside, and then they repack in the wake of your arm. Everything else is completely unaffected, right? You just locally yield that packing, and then it re-solidifies, right? Similarly, in these granular hydrogel packings, what we do is we built a 3D printer in my lab where you have an injection micronozzle that's mounted on a position-controlled stage, plunge that into the hydrogel matrix, extrude cells from the tip, trace out any 3D path you want. As that nozzle moves, right, it's extruding cells along its path, it's pushing the hydrogel grains out of the way, and then they repack around the injected cells and hold them in place. So this is a form of 3D printing. We were inspired by related approaches people have developed to 3D print mammalian cells and tissues and organoids. We've now hacked this approach to 3D print microbial communities and microbiomes, right? So we're having a lot of fun engineering designer microbiomes. If you're interested in engineering a microbial community of a given structure and a given composition, come talk to me. We'd be interested, right? Of course, we had to 3D print the prints and shield. These different colors are different layers, right, inside the media, because otherwise I wouldn't have gotten tenure, right? But we make these out of bacteria. So we're having a lot of fun engineering microbiomes, but for the purposes of studying this question of how do the cells collectively migrate through crowded spaces? Can they collectively migrate through crowded spaces? We 3D printed the simplest possible thing you can imagine. We 3D printed a cylinder of densely packed cells, right? So this cylinder is much longer than what you see here. This is just a small field of view. You're looking at a slice through its midplane, and the cylinder has been 3D printed within one of these hydrogel matrices. So it's completely surrounded by this hydrogel, right? The entire width of the screen here is thousands of cell body lengths. So this is really large, and it's surrounded by this hydrogel matrix. So the question is, how is this initial distribution of cells going to spread through the surrounding matrix? Any guesses? Oh, wait. Perfect. Thank you, Ronnie. Yes, I just told you all that by tracking the individual cells, we found that they move through hopping and trapping, and you can model that as a diffusive process. So isn't this a beautiful example of a heat mass transfer problem? You have this initial cylinder that's going to diffusively spread outward, right? I completely agree, right? There will probably be some Bessel functions involved because there's always Bessel functions in diffusion problems, right? This is a clean way of independently tracking this diffusive process. That's what I thought, right? I was wrong. So I just gotten back in my chair. Tapa showed me this movie. I fell back out. This is what we saw. So again, the lighting is a little dim. I'll show you an end-on view. These things expand radially outward in fronts. They self-organize into these coherent fronts that just keep moving. Somehow they're coordinating their motion, and they keep moving over thousands of cell body lengths. They keep going until they hit a wall, right? somehow they're coordinating their motion, even though they don't know that there's neighbors around them, even though they're in this obstacle-ridden terrain, right? We can zoom in and watch the individual cells. They're indeed still doing this hopping and trapping motion, but there's some kind of a bias that enables them to collectively move. So how? How are they moving in these coherent multicellular fronts over tremendously large length scales? Well, the answer is chemotaxis. So I'll point out that by tracking the individual cells, right, we were actually able to discover something interesting about biology. It turns out that the way the individual cells bias their motion in these crowded spaces is completely different to the way they bias their motion in bulk liquids. And I'd be happy to chat about that afterwards if you're interested. But zooming out at the population level, what's going on is, well, initially we have this distribution of cells in green. They're embedded in this hydrogel matrix, which has nutrients all around it, right? So that's in orange. And what do the cells do? Well, they consume those nutrients, right? They locally deplete them, they establish a local gradient, and then they do what we do when we deplete our resources. We go elsewhere looking for more, right? They sense this local chemical gradient, they bias their motion up that gradient, that's chemotaxis, and so they move outward. As they move outward, because these fronts are so concentrated, the consumption of nutrients by this collective is so high that it continues to establish this local nutrient gradient that they continue to bias their motion in response to. So they just keep going, right? These are self-sustaining fronts. As long as there's more nutrient out there, they just keep moving, right? And so we can use the machinery of reaction diffusion problems that chemical engineers are so good at to model this problem, right? For example, we can write down a very simple equation for what the nutrient is doing. It diffuses with a given diffusion coefficient that we know, and it's consumed by the cells at a rate that's proportional to the local density of cells B, right? And so that's coupled to what the cells are doing. And so what are the cells doing? Well, they have their flux due to their motility, and also over long time scales, they have a net growth. In many of these experiments, they take hours, days, weeks. We have experiments that have gone on for months. So the cells at the end of your experiment are not the same that you started with, it's their progeny, right? But we'll neglect that for simplicity. Let's unpack this flux, this due to the motility. There's two components. One is a random component, right? That's this diffusive process, this hopping and trapping. The other is a directed component that reflects chemotaxis. So here, chi is what's known as a chemotactic coefficient. It's like a diffusion coefficient, but for chemotaxis, multiplied by the spatial gradient of this function f that describes how the cells logarithmically sense the nutrient around them, right? And I'll get back to that in a second. But together, these define what's known as a chemotactic velocity, right? This describes the velocity of these fronts. And so we can measure all these parameters, right? We can put them together. We can numerically solve this model, right? It works beautifully in describing the dynamics of these fronts in bulk media. It didn't do a very good job. It did a horrible job at describing what we saw in our experiments. So we scratched our heads, we thought long and hard until we realized that, of course, confinement changes the values of these transport parameters, right, in two ways. One is, as I told you, right, because these cells are confined, their diffusivity is diminished because of the confinement by the obstacles around them. And also because there's less free volume, the cells bump into each other. We visualize that. So they truncate each other's motion. So when we update these transport parameters properly to incorporate these effects of confinement, the details are in this paper here, we find really nice agreement with our experiments. And what's nice about this, you can do your typical non-dimensionalization of this, various non-dimensional groups pop out. It really kind of gives us lots of insight into the fact that confinement tunes the relative influence of motility and growth in describing how these populations spread through their environments. But here's an example of one of these numerical simulations of this model, and it captures what we see in our experiments really nicely. And in more recent work, we updated this model to now include the effect of extracellular signals that the cells secrete, for example, that's involved in producing biofilms. And so we've updated this model to capture what we see in biofilms. Some of you might be familiar with a phenomenon known as motility-induced phase separation, or MIPS, which is a really interesting phenomenon where motile collections of active agents can cluster together and actually phase separate in a manner that looks like spinodal decomposition. We've now updated the MIPS model to incorporate chemotaxis, and it turns out that chemotaxis can really profoundly change the phenomenology of MIPS. So that paper is on the archive here. But in my remaining few minutes, I'll just go back to this initial experiment, and I'll point out something that really bugged me for a long time. So we see these fronts in the experiments, right? We can 3D print them, we can study them, we can model them using this reaction diffusion type model. Okay, that was really satisfying, but something really bothered me about these experiments. Look at the shapes of these fronts. Again, it's a little dim here, but I think you'll notice that they're always smooth. They're always flat, even though these cells are navigating these incredibly crowded, obstacle-ridden environments, right? They're constantly bumping into things. Some cells are moving faster, some cells are moving slower. They're constantly being perturbed, and yet somehow they regulate their speeds to keep traveling as a smooth group. How is that possible? I come again from a fluid mechanics background, where typically where we have interfaces that are propagating due to a local gradient, right, like a local pressure field, you often have interfacial instabilities. Those interfaces are unstable, perturbations grow, and you get things like viscous fingering. Here, somehow, we're not seeing that. Why is that? Well, I told you that we 3D print whatever we want, right? So we said, let's 3D print our own perturbations. Let's intentionally force some cells to be ahead, some cells to be behind. No matter what the wavelength of the perturbation we 3D printed, no matter the amplitude, we always saw the same thing. So again, I sat back in my chair, Tapa showed me this movie, I fell back out. They always smooth themselves out. They always stabilize these perturbations, no matter what. And we see this in the simulations as well. And so through a lot of work, we realized that chemotaxis is somehow involved in this process. So the question is, how are these cells using chemotaxis to stabilize these large-scale perturbations to their overall population morphology? They don't communicate with each other. Great question. Well, they kind of do, but indirectly. These strains that we use don't secrete any extracellular signals that we're aware of. We tried to use them to come up with the simplest possible case. That's exactly what I thought. But they are communicating with each other indirectly through the nutrient field. So that's a great point. How do they do that? Let's go back to this chemotactic velocity. Okay, I mentioned that it goes as grad F, the spatial gradient of how the cells sense nutrients around them. So let's unpack that a bit. Typically, people simplify this and they just describe this function as a linear function. Okay, so chemotaxis goes as grad C, right? So that's this dashed line that you see here. That's not strictly true. People have measured these logarithmic sensing functions for various bacteria, right? So this functional form here is one of the most experimentally validated functions that we use in our work. But the functional form doesn't really matter. That's what I'm showing you in the black line here. What matters here is that this function rolls over at high concentrations. It saturates. And this is a generic feature of many living and active systems that respond to external stimuli. Your ability to sense external stimuli doesn't extend over an infinite range, but it's limited. It has bounds to it, right? It plateaus at high concentrations, right? Why? That turns out to be the resolution to this entire puzzle. How is that? Well, in the spirit of linear response theory, we can unpack that grad F into two components. One is the typical grad C, right? The gradient of the nutrient. That's the driving force for chemotaxis, but it's modulated by the slope of this sensing function. It's like a response function, right? This describes how effectively the cells can respond to an external chemical gradient. And you'll notice that diminishes with increasing nutrient concentration, right? Your ability to respond to an external driver becomes less and less effective the more oversaturated you are. I really like coffee. Okay, and so my analogy for this is, well, let's say I've been drinking coffee all day. So I'm out here, right? And one of my friends texts me and says, hey, there's this new coffee shop that opened up down the street. You should come. The driving force for that is really high because all my friends are telling me to go there. But I'm oversaturated with caffeine. My response function is very weak. I'm not going to go to that coffee shop. I'm just too caffeinated. But let's say you pulled an all-nighter in the lab. You really need your caffeine, okay? So you're out here. Your response function is very high. But the only place that's open is the really gross gas station down the street with really gross coffee, right? The driving force is weak, but you really need your caffeine. You're going to chemotext to that coffee place, right? Because your response dominates. That's what's going on here, right? And so we can look at that through the simulation. So these are two contours of nutrient concentration. You will notice that at these peaks, these leader cells have a steeper chemical gradient, a nutrient gradient, right? The contours are closer. These followers have a shallower nutrient gradient, right? As you might expect, right? So these leader cells experience a stronger chemotactic forcing. So they should move faster. But that's not the end of the story, right? If that were the end of the story, then those bumps would move faster and this population would tear itself apart. You'll also notice that these leader cells, because they're jutting out, they're exposed to more nutrient. These ones are exposed to less nutrient, they're hungrier, right? They're less oversaturated, and so the response is stronger. And because those follower cells have a stronger response, we can quantify this. We can quantify how much faster do these move compared to those leaders. And the time scale it takes for these to catch up is exactly what establishes the time scale over which these large scale undulations smooth out, right? And so what we thought was really interesting about this is that this simple quirk of how individual cells sense and respond to external stimuli has profound implications for keeping the entire population together, no matter how much you perturb it. And more recent follow-up work, we came up with a theoretical model generalizing this idea to other forms of active matter that respond to other forms of stimuli. So we think this might be a general principle in living in active systems. And so with that, I'm going to end by just pointing out that everything I told you about thus far was for the case where the pores in these hydrogel matrices were large enough to let the cells swim through, right? But as we make the pores smaller and smaller and smaller, there comes a critical pore size comparable to the body size, right? Where now, even if the cells are motile, they're stuck. They can't swim through. In that case, they can only consume nutrients, grow, divide. Their turgor pressure is large enough to squeeze open the pores and push their daughter cells into the next pores, and so on and so forth, right? So now this is the purely growth limit. And in that case, when we do the same cylinder experiment, right, instead of seeing this smoothing that we saw something completely different. These grow into broccoli, right? They grow into these beautiful, large, multi-scale fractal-like structures that look like broccoli. In fact, we've characterized this and shown that they're fractal-like at small length scales, but to use an analogy of broccoli, there's a characteristic largest wavelength like the floret size of broccoli, right? And that seems to be consistently reproducible in all experiments, right? We found that it's independent of the hydrogel pore size, the hydrogel deformability, the nutrient type and concentration, the initial conditions of the colony, and we tested many different cell strains. We tested Vibrio cholera, which is a biofilm former and a gut pathogen. We tested Pseudomonas aeruginosa, which is a soil bacterium and a lung pathogen, also biofilm former. We tested kombucha, the stuff that's in kombucha. I don't know if any of you like kombucha, but there's often a sludge at the bottom that's called a SCOBY. That's a biofilm. We tested one of those strains for fun. We always see the same thing. So this appears to be a generic morphological instability exhibited by bacterial colonies growing in 3D. And I won't go into the details of this, but it turns out that the physics of this is very simple. And it reflects a fundamental difference between growing in 3D versus growing on a 2D surface. If you're growing on a 2D surface, right, you can always access nutrients from the third dimension. Whereas if you're growing in 3D, at some point, inevitably, you're going to get to a threshold size where the cells on the interior are depleted of nutrients that they need to grow, right? And you can do a very simple balance of the diffusion and the consumption by the cells to estimate that length scale that nutrients penetrate before they're depleted. And it's actually quite short. It's only 10 micrometers. That's just a few cell layers. And indeed, through imaging, we found that growth and metabolic activity is just localized to a thin surface layer at the surface of these growing colonies, right? And so what we did is we constructed a model of these growing colonies, treating them like active fluids, where now the fluid pressure is related to nutrient availability. We performed a linear stability analysis of that problem, and we found that there's the most unstable length scale that's set by 20 times this nutrient penetration length, and that captures exactly what we see for this floret size in our experiments. Okay. And so with that, not only do we find this in the lab, but when we look at nature, for example, colonies growing in the ocean or in bioreactors, we see similar phenomena, and we're continuing to explore the implications of this. For example, Mira, Alejandra, and Carolina have been studying how if you have a mixture of two different strains in these colonies, they demix. They demix in these florets as they grow. So maybe this has implications for maintaining genetic diversity in mixed colonies. But with that, I'll end here. If there's one takeaway you take from this talk, it's that being forced to live in a tight spot fundamentally changes how bacteria behave. Using imaging, using tools from biophysics, we found that the way individual cells move is completely different from what the textbooks told us. The way collectives move is completely different from what we expected. And the way collectives grow is completely different and provides us with rich sources of new inspiration for future work. This is just the tip of the iceberg. I hope today I've told you how you can observe a lot just by watching. We have other studies where we use this approach to study bacteria, as well as polymer solutions, colloidal dispersions, hydrogels, and ultimately we're excited to use these kinds of studies to bridge the gap between studies of soft and living matter in uniform idealized spaces and their applications in more complex and crowded environments in the real world. With that, these are the people who make all this work happen. Thank you all for listening, and I'd love to hear your questions.
Incredible. Like Sujit, we must have some questions. Please raise your hand for questions. Yes.
First of all, thank you for the amazing talk. It's really interesting work.
Thank you.
I think I have two questions. First one is, did you also just try some, let's say, more complicated or complex printing patterns that you had, for example, 2 cylinders and fronts were meeting or something like that?
Yes. Okay, I don't have a slide for that, but it's in our 2019 biophysical paper. We did exactly that. So we did, do you mind if I write on the board?
Absolutely.
Okay, we did exactly what you just said. So I told you.
Try like 20 markers before you get. Oh, great.
It worked on the first try. So I told you about 1 cylinder that expands radially outward. We also did 2 cylinders. And what's interesting there is, well, you can imagine if you have 2 cylinders that are really far apart from each other, right? Well, they're not going to see each other. They're just going to do their own thing. Okay. Bring them closer and closer and closer. Actually, these fronts move and eventually they collide and they annihilate. Bring them closer and closer and closer. Eventually, there's a critical length scale at which now the gradient established by one cylinder influences the other. And these fronts don't propagate inward anymore. They just move outward. We've also found that for circles. So we 3D printed circles where, again, you know, above a critical radius or diameter, right, you have fronts that go inward and outward, but below that critical diameter, the cells on the other side see each other, and so they no longer do that. So they're interesting things. We've also done lattices to look at interference, all sorts of fun things. Yeah, I tried to convince one of my postdocs to 3D print Imperial College, but
yeah, that's really good. Thank you. And my second question was, I was wondering if this hopping and trapping, or let's say whether the trapping isn't in some way just a concatenation of a lot of unsuccessful run and
tumbles. Yeah.
Or is it really that they pause for some seconds and then they just do the next? Or
no, I think you're absolutely right. I think it is exactly that. basically, and so we've thought about this quite a bit, and I think there's still quite a lot of work to be done. I'll show you just one cartoon. So we were oh yeah, here you go. So we were inspired actually by, so in our experiments, we can see that they're sitting there constantly, the bundles are constantly rotating. We don't think they're changing their behavior. We think they're still trying to perform tumbles, but now they're confined. And so we were actually inspired by models people have constructed for large polymers that are trapped in porous media. And in that case, for example, if you have a polymer that's stuck in a tight spot, okay, and just due to KBT, due to thermal energy, it's taking on different configurations. Well, you can notice that for this polymer to escape this tight spot, one of the two ends needs to be near an exit for it to transition out, right? So there are only a certain number of configurations that enable this thing to escape. All the other configurations keep it stuck. So you can count those, right? And in the spirit of statistical mechanics, right, this looks like an entropic trap, right? And so you can write down a kind of confining free energy well, and I'll just move this, if I can move this, yeah. And you can write down essentially an Arrhenius type of expression for how, just thermally activated, right, this polymer, how long it takes for it to escape this tight entropic trap. So we said, well, in our experiments, it looks like the bacteria are doing a similar thing, right, where now a cell is stuck, it's wiggling around, it needs the right orientation to get out. So there are only certain orientations that enable it to escape a tight spot. All the other ones keep it stuck. And so inspired by that, we constructed an entropic trap argument, and then you can think about how this propagates to a disordered landscape, because not all the traps are the same. And there are predictions that emerge from this that we were able to verify in the experiments. So that was a long-winded answer, but I think in short, we think that they're still trying to do tumbles, but the fact that they're confined in these tight spaces reduces the number of configurations that enable them to actually escape.
Really interesting talk. I have two questions. Maybe I missed something. One is about the transition between the two types of behavior, the run, tumble, and the kinds of things you have done. Were you able to probe like what happens in the intermediate zone, so to speak?
Not yet. That's a great question. So again, everything I talked about, today for this hopping and trapping is in the really highly confining limit, where essentially, if this is a cell and in bulk liquid, let's say the mean run length, is something like this. Well, we were probing, before it reorients, we're probing the case where the confining dimension is much shorter than this mean run length. And then you see this hopping and trapping. Now so let's call this the confining length scale, right? So in this case, L confining is much shorter than the run length. You can imagine the other limit where the confining length scale is much larger, that you just recapture run and tumble. I suspect that there will be interesting things in the intermediate regime where now they're comparable to each other. We know that these cells, due to hydrodynamics, for example, like to hang out at surfaces. And so one might imagine that there would be other interesting modes of motility when these length scales are comparable. That's something we're interested in doing, but we haven't explored that deeply enough yet. It's a great question.
The second question is about chemotaxis, rather than at the population level, at the single cell level. What implications do your studies have for chemotaxis at the single cell level, including studies on mutants and such?
Yeah, well, that's a great question. That I could go on for a long time about that. So I'll keep it short. So two things in particular that I think are particularly interesting that emerged from our work. One is by tracking the individual cells. So typically in the textbooks, what we read is how do these cells like E. coli perform chemotaxis in bulk liquid? Well, what they do, so they're doing this run and tumble. Let's say I'm a cell that's doing run and tumble, and I sense a chemical gradient. So I sense that there's more nutrients over there. So as I run, because the cell body is small, it can't detect a chemical difference across its body. It has to do that through moving. So it's moving. As it's moving, it's detecting that conditions are getting better. So it's going to suppress tumbling. It's going to elongate runs in this direction. If it's going down the gradient, it's going to promote tumbling, shorten runs in this direction. That was the paradigm of how we think about chemotaxis. There were some studies that were performed about 10 years ago where people found that in bulk liquid, there's actually another mechanism that the cells use to bias their motion, but in bulk liquid, it's very weak. Its contribution to the overall flux is less than 30%. And that's known as directional persistence, where a cell is moving up the gradient, okay? It senses that conditions are getting better. Not only does it elongate that run and, suppress tumbles, but it also modulates the number of flagella that become unbundled, which influences how much the cell body reorients. So it reorients less when conditions are going better, so it's more likely to keep going up the gradient, and so on and so forth. But the contribution of that mechanism was very weak in bulk liquid. So the question then is, why should these cells use two distinct mechanisms to bias their motion when one seems to be doing the job? What we found by tracking the individual cells, so the details are in this paper here, that, and the question about the two cylinders, that's also in this paper. So what we found is that in crowded media, this primary, quote, primary mechanism of biasing the motion doesn't work, because even if you want to elongate your hop, you're going to run into a wall. If you want to shorten a hop, your flagella remain bundled, you can't shorten. And we directly showed that. But this other mechanism is what does the job. So we think that having these distinct mechanisms to bias the motion enables these cells to perform chemotaxis in more diverse environments. So that's one kind of interesting biological feature. The other, and I'll just say this very quickly, just in the interest of time, is, we measured, for example, the trapping durations, the distribution of the trapping durations, right? The duration over which an individual cell is stuck. They're power laws. So here are just some of the data where these are log axes. You see these beautiful power law tails. And so what's interesting that, these trapping durations can be as long as tens of seconds, right? For these cells, again, when they perform chemotaxis, they can only remember the conditions over a certain time scale. So cell is moving up this gradient. It integrates whether conditions were getting better or worse over a memory time of around 5 seconds. People have measured that. And so imagine now if a cell, 5 to 10 seconds. So imagine now if a cell, if the porous medium is so tight that the cells are trapped for longer than that memory time. Will they forget and not be able to perform chemotaxis at all? That's one implication of our work that we've been trying to test, but no results there yet. So those are two. I hope that answers your question.
I think it's the same question again, so it's repeating, but what do you think speed between a run and a hop?
We didn't measure any difference in the speed.
Okay, but do you think that it's related to because you have a more a constraint on the run bacteria, or there's the BFN is different and this fail tumbles actually make other BFNs having to pull the one that was a tumble?
Can you say that?
The bacteria failure motor is the ones that want to make a tumble.
Yeah.
Still that one gets stuck.
Yeah.
And the others are still trying to pull it.
That's what we think is going on. But we're trying to probe that more deeply. All of our experiments suggest that picture, but we're trying to test that more directly. That's a good question.
Can I ask a question? Okay, thank you. So in the last example that you showed with this the rough object, I mean, if this is, I was just thinking about similar, let's say, other rough interfaces, like let's say in capillary inhibition.
Right.
Where you do there, because typically here you would have the long range correlation all along the interface. So it means that if you, let's say, disturb one point of the interface, it will be felt everywhere else. That's critical for a pinning process.
Right.
In addition, this is because of surface tension.
Right.
So what is the mechanism here for this long-range correlation?
Great question. So, and again, I appreciate that question because this is essentially an active analog. The way we think about this is an active analog of, what's often referred to as Mullen-Sukruka instability in growing kind of inorganic films. You mentioned in capillary inhibition, you get these rough fronts. And so here, what emerged from, the way we think about this and what emerged from our analysis is very simply the following. So let's say you have you have one of these cylinders initially flat, and let's say, just due to random fluctuations, you get a little bump. So these cells are now out ahead. they have more access to nutrients, right? So they can grow faster, and this bump will elongate. That's a destabilizing mechanism here. But not only do they grow outward, but they can also grow inward. They can fill in these gaps. And that's a stabilizing mechanism, right, that operates. And so actually, when we perform just our simple linear stability analysis of this active fluid model, where the important feature is that the growth is coupled to nutrient availability, right? So if you just look at the growth rate of perturbations of a given wave number, right? Our dispersion relation looks something like this. Okay, so these
it should be smooth at 0 wave number, yeah?
Yes,
This should be smooth.
exactly. And so positive means it's destabilizing, it's unstable, and negative means it's stable, right? And so this is what gives this magic kind of most unstable length scale. And this emerges from the competition between essentially this destabilizing mechanism of fingers growing outward in response to nutrient availability, but the stabilizing mechanism of being able to fill in the valleys. I hope that answers your question, but
Yeah, did you try to look at the velocity of this front end, like as a function of wave number? Let's say, so what they do, let's say in similar systems, they will look at the power spectrum of the velocity.
We did, yeah.
You did, so there is okay, so it is enough also, so you saw a decay. Right?
It is, yeah, we absolutely did. However, what's interesting about this, so you mentioned.
It is truncated though. Because
It is, absolutely. And what's interesting though is, you mentioned pinning, depinning processes, which people have in non-living systems have modeled using, for example, the KPZ equation, vertebrae design kind of rough.
yeah, this is when you grow up or down, yeah?
Exactly.
Yeah.
Right. And so what we found actually by looking at those power spectra of the fluctuations is, and looking at the roughness exponents, is that these growing cylinders follow what's called family-Vichak scaling, dynamic scaling. So they are indeed fractal at small length scales. However, the roughness exponent and the growth of that roughness exponent is larger than what you would typically predict from KPZ. So they appear to be rougher than we might have thought of for other non-living systems. We don't know why. So that is still an open theoretical question. Yeah. But we have, lots of characterization of those power spectra in this paper, and lots more.
There is one modification of that model where it takes into account memory effects and then it shows that it's a larger, but that's another question.
That could be it. I mean, there's a lot to do there. It could be some reflection of quench disorder.
Yeah.
So, yeah.
Yeah, just following on stability analysis, can I ask you what is the assumption you are using for the basis for the base state, and how many eigenvalues you are tracking there?
So, okay, I think we should, that is probably a longer answer that we should talk about afterwards, but I'd be happy to, yeah.
You're in the game.
Yeah, exactly. Yeah, I think there's a lot to talk about there. Yeah.
One last one, go on then.
I hope it's much quicker. You showed us that this one for experiments, and you said that you can differentiate between open trap mechanism and as opposed to that just a simple gas, oxygen. So if you look at the arrival time distribution, can you actually then establish what is tortuosity from the oxygen experiment and use tortuosity to have better prediction for open trap part? Because the effective diffusivity also depends on the tortuous paths.
Yeah, that's an interesting question. I think we should talk more about that afterwards as well.
Versus oxygen, and then use that.
Yeah,
for the,
it's an interesting thought. The one thing is that the oxygen, for example, and those small molecules don't see that solid matrix. So their so the tortuosity that you measure for a small passive tracer versus one of these cells would be very different. Yeah, and so, but that's an interesting point. We haven't directly measured those tortuosities, but we have the data. So, I'd be happy to share those with you if you're interested.
I have one about bacteria because I don't know anything about it. I expected to see it multiplying.
Yes.
How come you only showed it multiplying when it was really stuck within the confines and couldn't move forward, and then it multiplied?
That is an excellent question. That's a fantastic question, and that goes to a question of timescales. Okay, and so, for example, So in short, the doubling time of these cells is around 1 hour, half an hour to 1 hour. Okay. For a lot of the experiments that I showed, especially with motility, those timescales are much shorter.
Right.
Whereas, for example, this experiment is going for tens of hours. Some of them go for even longer. And so in those cases, you will certainly have proliferation. And
that's what you meant, that the one that makes it to the end is not the one that started.
Precisely.
Right.
And so that actually, you can quantify that. So, you have growth terms and you have motility terms. And so one interesting feature, again, is that if they're not really confined, then their spreading is set primarily by their motility. And they spread very quickly over short time scales, over which proliferation, growth, doesn't really play much of a role. But then as you confine them more and more, you slow down that motility, and then growth, and so the time scales are longer, and then growth is playing an increasing role in determining their spreading dynamics. So there's an interesting competition of time scales that we've analyzed quite a bit in that second paper. Yeah, it's a really important question. Exactly. Yeah.
All right. I think should we one more or we're concluding? Okay. I'm getting it. Can we just come afterwards?
I'd be happy to chat afterwards. Yeah.
Yeah. Okay. Well, shall we thank Sujit for the wonderful.
Thank you.
For your next appointments.
I'm game for anything.
That was brilliant.
Oh, thank you.
Thank you. I appreciate
Thank
it
you so
very
much.
much. Although it's not my field, I was like, yeah,
Oh, well, thank you.
fascinated. Yeah.
That's very kind of you. Let's see, I imagine I have to do some things
So
here.
we need to stop the recording and to make sure that it's.
Feel free to.
I don't know what.
I don't know anything about Teams, so I'm going to defer to.
Whatever it is that you want to collab, would you say just connect yourself and I will sort out the rest.
Good to see you.
Oh, it's not my
Hey.
Hey,
Thank you. Yeah, exactly. I'll see you.
Very nice. I'm Brian.
Yeah, we talked about something before. I'm not sure.
I want to just have the PC. Where is the recording starting?
I think I started it from my phone.
So you can stop the recording.
Yeah, I stopped.
You stopped it. Okay, so then that's fine.
Yeah.
So then leave.